Hello everyone, and welcome to this audio podcast. This audio podcast, as part of the Percolation. This audio podcast is part of a Bostic's Torpedo and the Specials. This audio podcast as part of a Spectacle and News Flash, and this audio podcast is part of a Storm Robotics production. So through this audio podcast, a look at more some of the more advanced sort of laboratory investigations. Some of the more advanced laboratory investigations in looking at the diagnoses of osteomyelitis, and staph aureus or S aureus. So, in addition to lab bench biochemical investigations, which presumably identified bacteria based on phenotypic observation. Automated molecular investigations are performed or can be performed to sort of pinpoint to pinpoint to pinpoint to pinpoint staphylococci to a genotype to at genotypic level. This is particularly important. Is particularly important when S. aureus is a causative agent of infection due to MRSA associated risks. Um, Additionally, sort of matrix assisted laser desorption or ionization, a time of light, a time, a time, sorry, ionization time of flight mass spectrometry, thus being an automated molecular genomic based investigation, can be used to detect to detect bacteria, including MRSA. Particular agar plates, agar dishes, in context of laboratory investigations, such as moldy tough MS, identifies bacteria can help identify bacteria by can help identify bacteria by producing peptide fingerprints of unknown organisms, for example, in amongst patient or recipient samples, and comparing them to peptide fingerprints found in proteome sort of databases. Moldy tough agar plates are also can also thus sort of offer a rapid and sort of less laborious modality to characterize bacterial identification than, for example, standard lab bench investigations. Additionally, molecular and genetic investigations, including whole genome sequencing, DNA microassays, and real-time polymerase chain reactions or PCRs, are used to detect specific bacterial strains. And by targeting specific genes, this can encode by targeting specific genes, specific or particular genes that this encodes the this can help encode the elongation factor of bacterial species, which then can be identified at at genus and species level. That draws this audio podcast to a close. This audio podcast as part of the Percolation. This audio podcast as part of the Bostics and Torpedo and the Specials. This audio podcast as part of the Spectre and Newsflash. And this audio podcast as part of the cases and evidence-based. And this audio podcast as part of a Storm Robotics production.